Okay, guys, today's unit is all about genetics and heredity, and I believe this is Unit 7, Section 2, Exploration 1. Okay, and today we'll really focus on Mendel and heredity. And so, just looking at all this, thinking back to a historical perspective of how we used to look at genetics, is really known as pangenesis, which basically means whole birth. So pan being the prefix for meaning whole, and genesis meaning birth. And there was this original kind of thought that basically organ cells shed these little things called gemmules, or these little pieces of material that were carried through the bloodstream to the reproductive organ, where they basically over time accumulated in the gametes. And so that was kind of the original idea, this pangenesis. Then there was this other theory called preformation, and really that was the discovery of the sperm and the egg that led to this idea, and that basically organisms come preformed usually from the sperm, so that the sperm was the more meaningful piece of genetic material. And then Gregor Mendel came around, and he's who we know as the father of genetics, and he is really responsible for what we currently understand about inheritance and patterns of inheritance. And he figured all this out by using peas, or pea plants. And so, why did he use these pea plants? Well, the main thing is that they were available at the time, and they're very easy to maintain, they're very easy to grow, and that there are many varieties or traits across the different species that are very easily visible. And so that's what we call a phenotype, or an outward appearance. So phenotype is the outward appearance. And I'll just write that down so that you guys can see. And so, phenotype, outward appearance. Sorry about my handwriting, guys. You know it's pretty bad. It doesn't get any better on an iPad. And so if we look at the pea plants, there are lots of different varieties. So first we can look here at height. So you can either have a tall pea plant, or a dwarf pea plant, or a short pea plant. You can have pea plants with purple flowers or white flowers. You can have them with their flower position that changes, and so it could either be at the very tip of the plant, or it can be more axial, meaning it comes more from kind of where the stem and branches meet. And then if we look at, so that's just the plant itself, and then if we look at the actual pea itself, you can either have different colors. You could have yellow or green peas. You could have round seeds, or you can have wrinkled seeds. You could have green or yellow pods, and then finally you can have smooth versus constricted pod shape. And so basically what that is in the smooth case, you don't have the little kind of tightening or shrink wrapping around the peas themselves, and so it appears smooth rather than constricted, which is what that guy looks like. The other thing is that pea plants self-fertilize, and so basically what does that mean? It means that the plant has both male and female parts, and it can reproduce by itself. And so if you look at this flower over here, you can see that in here is where you have the actual egg, and then the sperm is located at the end of these anthers here, and so that sperm can fertilize the egg in the self-pollination process. And so Mendel used these techniques to determine whether a plant was what we call true breeding. Okay, and so that's when a plant produces the same offspring generation after generation. So if it was, say, a purple flower, you got a purple flower in one generation, and a purple flower in the next generation, a purple flower in the subsequent generation, and so on and so forth. And the other thing he did was cross-fertilize. And so he basically used a paintbrush. So you can see the tip of the paintbrush here. And basically he touched the tip of that paintbrush to the tip of the plant over here. And then he took that sperm material in that pollen and transferred that to the egg over here. And then if you look at this picture here, he actually prevented any self-fertilization from happening by basically cutting off the stamens. And so that's these guys. Those are the pollen-producing structures. He cut those off so that this purple plant didn't create any pollen. And therefore, the only material that was transferred to the purple plant was the material from this white plant's pollen. And that was it. And so he took that sperm, dusted the carpel or the egg producing structure here at the tip, and then cross-fertilized the white and the purple plant. Okay, so before we get even more into kind of monohybrid crosses and ultimately dihybrid crosses, there's a few things that you need to know from a basic terminology standpoint. And so the first thing is alleles, okay? And so when we talk about alternative forms of a gene, we're referring to the alleles. So for the example of the purple and white flowers, there's clearly an allele for purple and an allele for white. Each organism has two alleles for each trait. You get one from mom and one from dad. When you have two alleles that are the same, meaning you have either, and so alleles are represented by letters, okay? So the letter really doesn't matter. There's no right answer for a letter. Just what you need to make sure is when you're representing a particular trait, so for example, color, you need to represent it with the same letter. When you have two alleles that are the same, you represent that with either two capital letters or two lowercase letters, and that's what we call homozygous. Homo as a prefix meaning the same. When you have two alleles that are different, that's referred to as heterozygous, and the only representation you have there is a capital letter and a lowercase letter, okay? And so this lowercase and uppercase P, in this case, are representing the purple and white flowers. So notice P was chosen because P for purple, but the white is also represented here by a lowercase p. So the dominant allele, okay, is the allele that is represented outwardly, even if you have one allele. Okay, so you only need one of the two alleles in a heterozygous individual, so like this. You only need one of those to be present to have that, to show that dominant allele outwardly. The recessive allele, okay, so that would be the little p in this case, that does not appear in the heterozygous case, but it does appear when you have homozygous. Okay, so you need, in order to display the recessive allele, you have to have two copies of it, one from mom and one from dad. Okay, so let's go over a little bit more terminology. All right, so hybrids, basically that's the offspring of two true breeding individuals. And so in this case, it would be a true purple and a true white. Okay, and a hybrid would be what you get when you cross those two. The P generation is the parent generation. The parent generation always refers to the true breeding plants. Okay, so in this case, that would be capital P, capital P, lowercase p, lowercase p. Okay. The F1 generation are the initial hybrids. Okay. And when we look at those, these guys are all going to be that. And they all appear purple because they have one copy of the dominant gene, which is that capital P. The F2 generation are products of self-fertilization of those F1 plants. Okay, so this is the F1 generation here. If you allowed every one of these guys to self-fertilize, just meaning fertilizing with themselves and no one else, then what you basically always get is what we call a 3-to-1 ratio, and it's 3 of the dominant phenotype to the recessive. Oops, sorry guys. All right, so then you have the parental cross. So I just want to do this out with you guys so that you can see how we arrived at those heterozygous individuals in that F1 generation. Okay, so this is what we call a monohybrid cross. Okay, so when we have a monohybrid cross, we're looking at one allele. Excuse me, actually one trait, not one allele. Okay, so you're looking at one trait and a monohybrid cross. In this case, what we're looking at is the color here of the peas. So we have a green and we have a yellow. So the yellow is represented in its pure form as YY, and so the way we represent that here is capital Y, and then the lowercase is the pure green. Okay, and so this represents the recessive trait. So the green is going to be recessive, and the capital Y, or the yellow, is going to be dominant. Okay, so what happens when you cross the green and the yellow? So pure green, pure yellow. What you end up getting is as such. And so the way that I filled that out was basically I just combined this Y with this Y, and this Y with this Y. So where do these intersect? Okay, and I'll just draw that better over here. So basically, it's what intersects in that column. Okay, and then you write those letters in those columns. So you can see here, when we mate basically the pure green and the pure yellow, what you end up getting is 4 of the heterozygous phenotype. Okay, when you are heterozygous, you basically end up displaying whatever the dominant phenotype is, right? So everybody here has that capital Y. We know from this picture over on the top right here that these guys are the dominant phenotype, therefore giving you yellow peas. So you end up getting 4 yellow peas. Notice though that these are now no longer true breeding homozygous for that trait. And so now you end up getting heterozygous. So they are carrying that genetic information for the recessive gene. Okay, so what happens then if you allow those F1 plants that we just saw to self-pollinate? And so the F1, remember, you have the heterozygous phenotype. So let's set up the monohybrid cross here. So you got a big Y and a little y. I definitely recommend that you use some sort of differentiation for your lower case letters. And so in my case, I add that little tail to them. And so here, what do you end up getting? So if we were to look at this, you actually have 3 genotypes, right? So you get 3 genotypes. All right, and what are those genotypes? You have YY, which is homozygous, and you have one of those. You get 2 YYs, which are heterozygous, and then you get 1 homozygous recessive, and this is homozygous dominant. Okay. So essentially you get 3 different genotypes. What else do you get? So you have 3 different genotypes, but that actually only represents still 2 outward phenotypes. Okay, so 2 phenotypes. All right. And the 2 phenotypes are still yellow and green. So how many of the peas are yellow and how many of the peas are green here? So the only one which is green is that little y. So you have what we call a 3-to-1 ratio then of yellow to green. And so why was Mendel so wildly successful with this? Well, the big thing is whenever you're doing genetics research, you should really only change 1 trait at a time. And that's exactly what he did. So he controlled cross-fertilization by cutting off the stamens, as we saw in that first experiment. And he was really able to control what was fertilizing, what plant was fertilizing what plant. He also verified true breeding parents. So he verified by doing multiple generations of that plant to find the purest purple or white or round or wrinkled parents. And then this was all very quantitative. He could count it all. He could see it very easily and calculate how many plants exhibited X trait and how many plants exhibited this other trait. And so it was quantifiable. He controlled it because there were really there's only 1 variable at a time, and he started with the purest forms of the plants. And so that'll do it for today.